Okay. So this is a joint work with Flávio. It's something I started some few years ago at Duke University in discussion with my advisor, Joe Lucas. But for many reasons, we never moved forward on this topic. So last year, talking to Flávio, we decided to propose this hidden Markov mixture strategy for the identification of overexpressed chromosome regions, which is a topic I have explored, some data I have explored in my PhD. So the presentation is divided in 4 main points. I basically will just introduce the data and then present the model and then show a real data application for cancer data. So we work with microarray technology, which has been used in many publications in the past years. We are particularly interested in this type of microarray here from Affymetrix. I show an image graph here in the log scale, where each spot it's called a probe, and they represent a fraction of a gene containing 25 nucleotides, those letters from the DNA. And the data is basically light intensities that you observe in the hybridization procedure. So when you see a bright spot, indicates the activity of the gene. When you see a blue spot, it means no intensity, no detection of the gene. But we are going to work with 5 different data sets. These are references. They have different number of samples. The largest one, 286, and another one here with 59. They represent different types of cancer, breast, ovarian, and brain. We have 3 breast cancer data sets. And the raw data should go through some pre-processing steps before being ready to use. Basically, we will apply log2 base scale and then adjust the measurements within and across the ships. Okay, in order to do that, we use a very popular technique, which is the RMA. So this is our goal. We want to identify chromosome regions associated with these high expression measures. So I highlight the high expression measures, why we are so interested on that. Overexpressed status suggests that the gene plays an important role in the cancer progression. This could be also related to anomalies like copy number alteration. And for example, in this study, Warren in 2007 proposed a method which basically classified the gene as present, marginal, or absent by choosing a threshold. If the intensity is larger than this threshold, then the classification is present. Okay, so it's something we are interested. Another part of the goal is the chromosome regions, how we are going to find those regions. Well, we use an alignment tool called BLAT, which finds genome positions with at least 95% similarity in the human genome. BLAT is a popular algorithm being used to find those sequences in recent releases of the human genome. It has a low computational cost, but also has some problems, which is not particular of BLAT. Also, other techniques also have some problems. For example, if the length is very short, you will have a higher risk of incorrect mapping. And inconsistencies, we may have probes being aligned to multiple locations and locations with multiple probes. But those cases here, we remove from our analysis because they are very few compared to the universe of thousands of genes that we are going to map and use. Okay? So you can check BLAT online for small searches at this address, and also you can download and install it in your computer through this website. Well, the resolution is not very okay. So I have here a histogram. I'm not sure if you can see, but basically we have histogram displaying a skewed shape and multimodal. And we are very interested in what happens in the right tail of the histogram, where the overexpressed values are located. Basically, our data mapped to the chromosome locations here in chromosome 4, they are as like a series with not a regular, with some distances between consecutive locations. These distances may vary, and we are going to use those distances to evaluate whether we have some Markov dependence between consecutive observations, okay? So we are going to use those distances as covariates in our model. So in the previous graph, we have 3,000 probes mapped to chromosome 4. And just to give an example, this is some descriptive statistics about the distances. The minimum is 1, the mean, the average 53,000, the maximum something above 3 million. So this introduced some computational difficulties. So in order to work with this, we decided to rescale those distances to the interval 0, 1 by applying the log, and then divide for the maximum distance, okay? So in our model, we are going to use those distances as covariates. You may try another transformation. This may change the inference, but it doesn't change the model that I will show in a few minutes. Well, given that we have this irregularly spaced series, basically we have something that is related to spatial statistics. So we applied this permutation test, I-Moran's permutation test, to compute p-values for example, a sample of 251 arrays across all chromosomes available for analysis, 24 of them, including X and Y. And all these p-values are below 0.05, indicating that we have a spatial association in the data. So the p-values are small, we have a spatial association, and I'm not sure if you can see, but here I have a histogram indicating the behavior of the distances in the new scale from 0 to 1. We have a group concentrated here, and another one in between 0.6 and 0.8. So those are going to be used as covariates. So starting to explain the model, basically we have n locations. L is the number of arrays. We observe this YIL, which is the light observed in position I in the array L. We have many arrays. We could model this value through this normal distribution with some mean xi, mean intensity, which is we would have to estimate. So in order to make a more parsimonious model and reduce a little bit our computational burden, we decided to use this result here. We basically choose xi to be the median of the arrays, and we are doing that because if you look at the location for a single location, all the expressions, you basically see a histogram with a symmetric shape for many locations we have tested, and basically this estimate would be something close to the median or the mean, okay? The model is, in order to model that multimodal shape and skew shape, we decided to introduce this mixture model with k gamma components and one normal component. The normal component has the largest mean, which will accommodate the overexpressed values. And basically, we define in our model this Markov structure with Q0 being a vector of probabilities when we don't have any Markov dependence across the array, across the sequence. And this transition matrix, which basically in each row, we have a different vector of probabilities indicating how can I move from one cluster to the other. Okay, given the previous cluster in the sequence. So some definitions for the distributions forming the mixture. And also we introduced this variable here, the zi, which is basically a vector and zik equals 1 if xi belongs to the kth components in the mixture. So this is the main part of the model. So xi given z has some distribution, which could be the gamma or the normal distribution. And here we introduce the Markov dependence. Basically, given the previous, the result, the classification of the previous value, we can choose, the model will choose between a multinomial distribution with some probability that is basically indicating independence, or this one, which is one of the rows of the transition matrix with the Markov dependence, introducing the Markov dependence. So in order to model the Markov dependence, we will do that for this probability row E here. So we introduce this WE, which is Bernoulli, and basically when WI is 1, we generate from this multinomial distribution, and when it's 0, we generate from this one, which is indicating dependence. Prior distributions, Dirichlet priors for the probabilities, and inverse gammas, and here we introduce this regression for the row I, and basically we use here, as I said, the distance, the rescale distance as the covariate with those betas, with some betas to be estimated, indicating how important is this D for this probability. Okay, so I introduce some few notations here. The Bayesian inference for this model, basically we define this VI here just to allow direct sampling from the distribution of the betas, which is directly connected with the W. VI larger than 0, W is 1. VI less than 0, W is 0. And we choose these blocks for the MCMC. We basically sample ZW, V, Q0, Q, the transition matrix, and the beta and phi, and also the eta. The connection between V and W makes this Markov chain non-irreducible. So in order to solve this problem, this part, we will use a collapsed Gibbs sampler where we integrate out V for the analysis. Okay. The sampling, the V, basically is truncated to be positive if W is 1, we generate from the normal distribution, and non-positive if W is 0. These are the complete conditional, the conditional for many of the parameters. We have a restriction for the model because we want the component of the mixtures to have some order. The gammas come first, and then the last one is the normal distribution, whose mean should be the largest one, okay? So we impose this to the model. So we propose from the non-truncated version, we're using rejection sampling. We check if this configuration is true to accept for the values generated, okay? The etas are sampled from Metropolis Hastings with random walk, Gaussian random walk proposal. And the more difficult step is to sample this ZW here, which has this full conditional kernel here. But we can factorize, use this factorization, which indicates that we can sample them through Bernoulli's and multinomial distributions in this order forward in time. Okay, so basically we will do forward sampling, but in order to update these values, we are going to calculate their parameters, update the parameters through a backward filtering. Okay, so we do backward filtering and forward sampling. So this leads to this algorithm which we call backward filtering, forward sampling, which involves a part here for filtering, which leads to the calculation of these parameters, and then we move forward with the sampling, okay, with this part here. In order to calculate the probabilities of each locations for being, or any sequence in those series on the chromosomes for being an overexpressed region, we basically take the mean of, for example, a sequence from 1 to S. We take the mean if the status of the K+1 component, which is the normal component, is 1. We basically take the mean of how many times that happens, okay, in the sequence. So our analysis focus is focused on this type of statistic. So the results for the cancer data. So we have 5 data sets: breast, ovarian, and brain. We are using all 24 chromosomes. I say 24 chromosomes because we include in the analysis X and Y. And we fix, there is some, the chromosomes are independent from each other. So the first position in each chromosome, we set W to be 0. In other words, there's no dependence between chromosomes. We will analyze them in a sequence, in a big sequence of 90,000 locations, but we have no distance to measure between chromosomes, so we set them to be independent, okay? So the choice of the, doing so, we can use the BFF step, which is a crucial step in our model, separately, which reduces a lot our computational burden. Okay, so we do the BFFS in our analysis for each chromosome within the MCMC. The choice of K, how many gamma components we are going to use. So we have tried K equals 2, but that leads to a normal component very wide, including values very low, considered very low to be an overexpressed values. So we have tried K equals 3, which has the results I will show in a minute, and we have tried K larger than 3. The results are pretty good with 3, and if we increase, this will only increase increase the number of parameters to estimate, so we decided to be parsimonious and use K equals 3, given that the results are, very nice. So prior distributions, we set this prior, encouraging the model to set observations in the gamma components, and so making it hard to classify some position as in the normal component. For the transition, those probabilities in the, for moving from one cluster to the other, if we have dependence, then we choose something like this. As you see in the main diagonal, we have the largest values. It's hard to move to the normal component, and if you are in the normal component, you can leave the normal component easily, more easily than entry in the normal component, okay, to be accepted in the normal component. So it's easier to move to the next cluster than to move to a cluster far away. Oh, and the other parameters are chosen based on some idea of the scale of the data, and we just choose some value in between. I will skip this part. So we move to the results. The MCMC is set to perform 15,000 iterations, burning 5,000. The chains are converging very well. And for generating the ATAs, we use a Metropolis Hastings, and we set those, we tune the proposal distribution to have acceptance rates around 44%. These results are the estimates, the posterior estimates and standard deviations in parentheses for the components in the mixture. So we have here the first gamma, second gamma, the third gamma, and the normal. So you see the uncertainty is lower for the mean than for the scale parameter. And when you look across the different data sets, we have similar results in terms of the mixture, the estimate of the parameters of the mixture. Well, there I'm showing the probability of each component estimated in each data set. So you see we have the weight, the probability of the fourth component, which will include the overexpressed status, is the lowest one, around 6%. And here we have the mixtures where you see here the normal component and the tail of the histogram. There is a histogram behind this plot here. And the gammas you see in yellow, green, and blue, and the density, the mixture density formed by the combination of them. Okay, we're using these weights. Here are the locations in the chromosome. I guess it's the chromosome 1. And you can see here in red those values considered the overexpressed values identified in the normal component in the mixture. Okay, here I have all 4 data sets. Okay, the same chromosome, and when you look at comparing different data sets, you check only those positions located in the normal component. If you check how many of them are identified as in the normal component in both data sets relative to the number of positions in the normal component in at least one of them. So when you do this this calculation, you see that breast cancer, the analysis comparing breast cancers has the largest similarity. So you see here at least 78% similarity. And when you compare breast and ovarian cancer, for example, you have around 70, and breast and brain cancer, you have around 60%. So larger similarity for breast, which is expected. They are represented the same type of tumor, and lowered similarity for brain. And surprisingly, we have the relationship breast and ovarian more similar than breast brain. Well, this is just an image plot indicating each data set, and you see these lines here in black, the probability of being an overexpressed region, okay, an overexpressed location. So you see here which locations have this overexpressed status, and you see the coherent pattern across this matrix, which indicates, which is an interesting result, especially when you compare breast cancer data sets with other types of cancers like ovarian and brain. You have some similarity between them, which is observed also in that table with those intersections. Here is only chromosome 1. Here the whole sequence with 24 chromosomes. Okay, so the sequence has 91,000 values. So the conclusions. Well, the main aspects of this study, we use the hidden Markov model in this application involving Affymetrix DNA microarray data. Our goal is the identification of chromosome regions. In order to do that, we use BRAT to align the probe sequences from the array to the human genome. And our data is irregularly spaced. We have distances between consecutive observations, so we use that information to define the Markov dependence, whether there is some dependence or not between consecutive values. In order to model, to cluster the values in an overexpressed status or not, we use the mixture distribution with gamma and one normal. The normal is the most important, which will include those overexpressed cases. And the main results, well, the model can discriminate well the regions with high expressions from the others. We can calculate those probabilities indicating they have overexpressed status. On average, we have 6% of regions with potential overexpressed clusters. Great similarity between data sets, breast and breast cancer, of course, and between breast and brain, breast and ovarian, which can be seen through the image graphs having a coherent pattern, and also with those intersections that are showed in the table. Okay, if you have any other if you want to know more details about this, we wrote a paper which is now in archive and is submitted to Royal C, so it's under review. Let's see what happens. But you can check here some details of everything I did, I presented here, okay? So thank you.
Okay, so thank you, Vinícius. So we have time for 1 or 2 questions.
Yeah, very nice. I got 2 questions. I like the applied results. There's several aspects of the applied results across the various cancers, including the characterization of the noise distributions, sort of makes sense. I mean, brain expression data from brain tumor is a very hard thing. So the 2 questions. The first question is the model. So you've got this Markov switching model on these sort of unit scale along the chromosomes, which sort of, I mean, implicitly indicates that you have to have the idea that the expression probes are sort of, you know, basically uniformly distributed across the scale and fill the scale. And don't you find pieces of chromosomes where there are big gaps, where there's just no probes?
You mean I may have probes disconnected from each other?
Or has your pre-processing BLAT or whatever it is removed all of that?
Yeah, like we may have that. I have checked some cases. Some cases I've checked, they are basically close to each other in the sequence, but we may have some pieces located in different regions, separate regions. But this may be cause for the problem of incorrect mapping, which is not the focus here. We didn't treat that part of the analysis. We basically use BLAT with that 95% similarity and just map to that location to see the dependence between values. But that may happen. You may have cases separate.
The second question has to do with the way you're pulling out your classification. You're thresholding the posterior probability of being from the expressed normal component at 1/2. What's the prior probability?
You mean here?
No, later on when you threshold, when you call it, you're thresholding at 1/2, right? The probability on a normal component. You're thresholding at 1/2.
Yes, yes.
Well, why 1/2? I mean, if
why not? Well, I have to have a threshold. I have to indicate what is a big probability.
Yeah, I mean, it's sort of calls,
it's above 0.5.
It sort of calls for some training data where you've spiked in some known samples, so you can actually calibrate. You know, you really need a decision theory sort of basis for choosing that threshold, right? But for that, you need data that you've controlled, and you know the results.
Yeah, perhaps if we would have some data to provide this sort of information, I will be glad to use. But in this case, we just have to pick a point. I didn't want to move too close to 1 or too far away from 0, so let's stay in the middle, so 0.5.
It's more than the Gaussian being the component with the highest posterior probability, because its probability needs to be higher than the sum of the other 3, so it's a bit more strict than just being the most likely one in the posterior, yes.
Okay, great. So let's thank both of our speakers. So before you leave, Flavio has a quick announcement.